Hey everyone, Sing here, back in the Minecraft lab. Today, talking about viruses. Now, viruses are super tiny parasites that need to take over another cell in order to survive and reproduce. Before we get into too much detail about viruses, let's turn on our super duper amazing microscope. There we go. And dive right into the structure of a virus. Now here in front of me, we have on my left a bacteriophage and on my right an influenza virus. Now both of these are two different types of viruses. Now there are a lot more than just these two types of viruses. Now the bacteriophage, hence the name bacteriophage, only infects different types of bacteria, and the influenza virus infects different animal cells. Now, both of these have in common, the one thing that both of these have in common is the protein capsid. As you can see, it is a protective shell like the cell membrane that protects the nucleic acid. And another term for nucleic acid is DNA and RNA. So both of these viruses on the cell inside have DNA or RNA that is either single-stranded or double-stranded. Viruses do not have organelles, as you can see from both of these viruses here. They do not have any organelles, no ribosomes, no cell membrane, nothing of the sorts. And now, you may be wondering, what is this magenta, purplish, pinkish looking membrane or layer around the influenza virus here? And this is the envelope or membrane envelope. It is an envelope that helps certain viruses that have them enter a cell fully. So they enter the entire cell. Now you're wondering, how can they do that? Are viruses so big? Are they so small? Or what size are they? So viruses are actually around 100 nanometers typically. That means they are 100 times smaller than a red blood cell. Now, these little pointy things on the outside, these jutting things out, are called antigens, and they attach to proteins on the outside of a cell, and they help to get into the cell. Now you may be wondering, why do these viruses want to get into cells? Now these viruses want to get into cells so they can reproduce. And to reproduce, there are two different cycles that they can do. The first cycle is called the Lytic cycle, and the second cycle is called the Lysogenic cycle. Now, let's go take a look at the Lytic cycle. So let's go back into the lab, and let's go, using our super duper amazing microscope again, let's go take a look at the Lytic cycle. So here we are, a bacteria, and so to explain both the Lytic and Lysogenic cycle, we'll be using a bacteria and a bacteriophage, or phage for short, to explain the process. Now, some of these processes are not exactly how other viruses get into the cell. Some of the viruses, not like the phage here, might go all the way into the cell or inject. It will make a lot of sense after I start explaining these cycles. Now, first, to start off, there are 5 steps in the Lytic cycle: attachment, penetration, biosynthesis, maturation, and lysis. Now, the first step is attachment, where the phage attaches to the bacteria. And that's pretty much the whole first step. That is exactly the whole step, where the bacteriophage attaches to the cell membrane of the bacteria cell. Now let's move on to penetration. So penetration is the second step. It is where the phage fully penetrates the cell membrane and injects its own DNA or RNA. Well, most of the time it's DNA. So it injects its DNA And different viruses do it in different ways. For example, influenza virus uses its envelope to enter the entire cell. But let's focus on phages. So it injects, it breaks through the cell membrane, and it injects its DNA. Now let's move on to the third step, that is biosynthesis. Here we are at biosynthesis. So as you can see, that there are actually, here again is the phage DNA, and there are protein capsids starting to develop. So in biosynthesis, the DNA enters the host cell, right, and it degrades the bacteria cell's DNA and replaces it with its own DNA and starts to make parts for more phages. So the protein capsid and the DNA. And that's all biosynthesis. Now let's move on to the next step, which is maturation, or maturation, I like to say. Anyways, now here we are. It's maturation. As you see, we're inside the bacteria now, and inside the bacteria, there are a bunch of phages. This is where all the phages, with the parts that you just saw, are assembled. So all the phages are fully assembled now. They're inside the bacteria cell and all fully assembled. And that leads us to the final stage / step of the Lytic cycle, which is lysis. That is where the cell bursts, destroying the cell membrane and killing the cell. So here we are. You see that the phages have burst through the cell membrane of the bacteria. They were inside, but now they're outside. And so once they're outside of that cell, they can go and infect and attach and repeat the whole cycle again. And as you can see, this poor cell cannot live without a cell membrane, so it dies. Now, that is the Lytic cycle. So the Lytic cycle is 5 steps: attachment, penetration, biosynthesis, maturation, and lysis, which is this, where it pretty much bursts from having too many phages inside of it. Now let's head back to the lab so that we can take a look at the lysogenic cycle. So here we go, once again, diving right into the lysogenic cycle. So here we have the phage again, and the bacteria. Now, the lysogenic cycle is a longer cycle with 5 whole steps, so 2 more steps. Now, actually, the first 2 steps of this cycle are exactly the same. So once again, it's attachment and penetration. So here we have attachment again. And so let's move on. Let's go take a look at penetration. Just a reminder, penetration is where it penetrates the cell membrane and it injects its DNA into the cell, like this. And then let's move on. Now this is a new step. This is called DNA incorporation. Now, this means the DNA gets incorporated into the cell. And this actually can be sitting there for months, years, and the cell wouldn't know. And in fact, the cell could split and reproduce, replicate, and the daughter cells, as you can see, daughter cells would also have the trait. So this part here, this magenta pinkish is the phage DNA, and this purple is the own cell DNA. As you just saw, that the daughter cells, the replicates of this cell, did have the DNA as well. So that's kind of the scary thing. And the cell doesn't know, this could be going on for generations and generations. And this kind of virus is called a latent virus. Now you may be wondering, how does this activate after being inside that DNA, maybe for months, maybe for years? how does it activate? And so it activates through stress. This is the next step. Stress. Where there are factors where it encounters stress. For example, the cell doesn't get enough food, therefore it goes into the Lytic cycle and starts biosynthesis, which I mentioned in the last cycle, where cells they degrade the DNA of the original cell and start producing parts for the phage. And another factor for stress is toxins, or just toxins in the environment, and that can also cause stress. So, like I said, we are back to biosynthesis once again. So after stress and after the DNA incorporation, we go back to the Lytic cycle and start biosynthesis once again. And a quick reminder of biosynthesis, where the DNA is of the original cell is degraded away, and phage DNA is produced, and protein capsids are produced to make fully formed phages in the next step, maturation. Right here, like before, as you can see, fully formed inside the bacteria. And to the last step, like last time, lysis, where the fully formed phages are so many, it's like a balloon, and it pops, and out comes tons and tons of phages that go and infect more cells. And this actually happens for every single daughter cell too. So back in DNA incorporation, that will happen to every single daughter cell, any cell that the DNA infects. Now, this has been the Lytic cycle and the Lysogenic cycle of viruses, and I hope you enjoyed this. This is going to be it for today. Thanks so much, and hopefully I'll see you again next time.